In this video, we will discuss about ubiquitination and de-ubiquitination and how this will affect in our immune system, or how this is related to our immune system. So first we will understand what is ubiquitination and de-ubiquitination process. So this process is related to recycling of the proteins not that are not that are either non-functional or not able to form their native structure, or because of any reasons which are the proteins are not able to perform their normal functions. So what happens first is that even enzyme even enzyme binds with the ubiquitin. This is a small protein that is usually present in every tissue of the in every cells in uh, in every tissue at different parts of the body. And this ubiquitin first binds with even enzyme that leads to activation of ubiquitin, and after that the conjugation occurs where even is replaced with E2 enzyme, and after that the E2 enzyme can interact with E3 enzyme, with E3 enzyme act as a ligase that can bind to the lysine of the protein substrate, or we can say specifically to the lysines of the protein substrate. And this ubiquitin then directs towards different kind of ubiquitination process, such as polyubiquitin, multi-ubiquitin, and monoubiquitin, as it is shown in the figure. In the polyubiquitin, different ubiquitin can be attached to in a series manner, whereas in multi-ubiquitin, can be attached either in both series and parallel, or in random manner. In monoubiquitin, only the one ubiquitin is attached. After that, this ubiquitin then act as a signal to direct them towards proteasome, where degradation 26S of the proteasome is where degradation of the substrate occur, or the protein. And after that, the stabilized substrate after that the degraded substrate can be recycled for the uh, formation of new proteins. Another way by which the proteins can be or not another way, the proteins that are able to form their native structure and can perform their normal function are considered as stable substrate, and they are not directed towards this proteasomal degradation. And these are the families of DUB, that is de-ubiquitination enzymes that can help the proteins for de-ubiquitination. So let us understand what are its impact on the innate immune system. In the innate immune system, different kind of signals uh, can uh, can be activated by de-ubiquitination, and that is responsible for the proper function of the innate immune system. As we know from our previous videos that there are two kind of immune system. One is innate immune system, another one is uh, one is adaptive immune system. So in the innate immune system is the primary response of the immune response of the body. So let us consider this example. In this, the virus is entered into the virus in this case the virus has entered into the cell, and this de-ubiquitination, this purple box shown as one of the enzyme of DUB, that is de-ubiquitination enzyme. And we can see that the purple box remove the envelope layer of the virus, and after that the genetic material gets translated. However, some part of genetic material can interact with the de-ubiquitination substrate, can interact with the de-ubiquitination substrate, and then it can go and finally activates the uh, IFN signal, that is uh, IFN signals. That is interferons, it will make the epigenetic changes to start expressing interferon genes and inflammatory genes. And this is shown here, that with the help of this envelope proteins and this genetic materials, this helps in this along with the de-ubiquitation process helps in generation of interferon genes or expression of interferon genes or inflammatory genes that leads to activation of IFN alpha, IFN beta, or interleukin 6, IL-6, and this will helps in expression of IFN AR at the cell surface level, and this finally it will help in generation of autocrine and paracrine signal. Autocrine signals are the signals which get activated, which get activated and release the chemical compounds that are used by the cells itself, same cells receptor. Whereas paracrine signaling is that uh, in which the chemical compound that are secreted by the cells can be used by the neighboring cells. So uh, this IFN AR, when it is expressed on the cell surface, it can detect the SARS-CoV-2 virus and release autocrine and paracrine signal, and this can be initiated whenever the cells get infected with the virus. This is how the de-ubiquitation or ubiquitation process is linked with the activating innate immune system. I hope you got some idea, basic level idea about the ubiquitation and ubiquitation and how they are linked with innate immune system. In order to understand these things in the detail, you can go through the references that are provided in the description and also mentioned in these slides. That's all for this video. Thank you for your time.